And good afternoon, everyone. I'm glad you were able to join us today for us to tell you a bit more about our new product, which is Jellagel. So the way we're going to work it today, just so that you all know, is that we're going to run through the full presentation. It's full of data, which backs up the product as well. So we'll have two speakers mainly. The first one will be Robbie, who is our inside sales manager. He is going to run through the initial part of the presentation. And I also have with us today is our founder and CSO, Andrew Mearns-Sprague. And he will be taking you through the data that we've been gathering on our product while using it in certain applications. So we hope that you get as much out of it as you can. With regards to questions, we're happy to take questions. Obviously, it would be great if you could all remain on mute while the speakers are speaking, just so we don't have any background noise. If you do have a question, there is a chat element to Zoom. If you could just please either write your question in the chat element or just let us know that you have a question, then I will come to you and we'll be able to answer it from that point then. I will stop the presentation, we'll try and answer your question straight away. Or there will be time at the end as well where we will be able to answer questions that you may have. So that's basically how we're going to run this webinar today. At the end, obviously you will have your contact through Florence at Clinisciences, who are our partner within the European region, and all of the contact details with regards to Clinisciences are on the back end of the presentation. So now I will hand over to Robbie Oliver, our inside sales manager, who is going to take you through the presentation. So thank you again for attending.
Thanks, Rob. I'm just going to share my screen, and this is, if there's going to be any technical difficulties, this is where it will be. Hopefully we won't have any. Hopefully you can all see that. A couple of you are on webcam, so if you could just give me a nod to say that you can see it.
So Rob, we're fine.
You're good.
Thanks. Excellent. So yes, welcome. Thank you all for joining us on today's webinar. We're going to run through our most recent product, which is Jellagel. It's a next generation jellyfish collagen hydrogel, and hopefully you will find today's talk interesting. So there are many reasons to adopt jellyfish collagen into your research. Many of them are very specific to our collagen. Firstly, they are prion and disease vector free, and we have shown that they are inert and significantly cleaner at microRNA level, and we've got data to evidence that as well. The product is biocompatible, and we've seen a very, very low immunogenic response in various testing. It's a batch-to-batch consistent product. If you imagine a jellyfish, there is not a lot there. They're about 90–95% water, and the rest is the sort of collagen proteins. Therefore, what you're getting in terms of the collagen content is a very batch-to-batch consistent product. When growing cells, we've seen sequence homology to types 1, 2, 3, 5, and possibly a couple of others as well. And it's because of this, the nature of the jellyfish collagen, it's being categorized as marine type 0. So, you know, you may be used to types 1 or 2 for your different cells, but this can be used across the board on a wide variety of cells. And as well as that, it is compatible with the existing cell culture methodologies in terms of self-coating, imaging, etc. So we've worked very hard in creating this product, and what you get is what's pictured in front of you. So there's the collagen solution and the buffer. They come in one pouch, which is then popped in the fridge. We've also got a custom cross-linker as well, which is stored in the freezer. but all of these products are actually used when you're creating the gel at room temperature. So there's no need for any cold rooms, cold pipette tips, ice, anything like that. So in terms of the user-friendly environment, it's very good, and we've had some great feedback so far. Also in the box, there is the protocol, which we worked very hard to simplify, and a user guide. So if you're using different quantities, hopefully you'll have all the information needed to make the gel. As mentioned, the protocol is a very simple step-by-step process. There is a quick video here. Again, I will try and play it, and hopefully it will give you all an idea of just how easy it is to form Jellagel.
Robbie, you might have to share that video separately. It's not coming through.
Is it not? Okay. In that case, yes, I will. How about now?
Yep, start it again.
There we are.
As you can see from the protocol video here, showing you exact volumes you can use, ways of administering it to the tubes. There's lots of detail on here, which is also on the paperwork as well. Very simple step-by-step video guiding you through it. So like Robbie said, there's two packets. The first one you just saw was the buffer and the collagen. And this one is the cross-linker, which will be added as well.
So yeah, that's the gel, and hopefully, yeah, it's a very easy process. And for anybody that has used any other hydrogels before, you know, it should be quite straightforward. Oh, hang on, what's going on here?
There's always technical issues.
Oh, yeah, there we go. YouTube had gone to the next video, World Engineering Day. That's for another webinar, I think. So yeah, next slide. So using Jellagel in the lab, the hydrogels can be formed in a variety of tissue culture coated plates. You know, everything, as mentioned previously, is pretty standard lab equipment. In terms of the stiffness, the values of this are very, very similar to Matrigel. So it should give you a very familiar feeling when you're using our product. I'm sure a lot of you do use Matrigel. So yeah, the feeling that you will get when using this will be very similar, and hopefully it will give, as I mentioned previously, a great user experience. Also, as I mentioned, it's prepared at room temperature, so there's no need for the cold rooms, cold equipment. And all of our products are in the ready to use vials, which again, for user experience is great. So we'll move on now to the data, and Andrew will take you through this. As Rob mentioned, Andrew is our founder and chief scientific officer. So I'm sure he will be very excited to talk you through some of the data which has only been released this week, I think. So we're very excited to be able to share it with you.
No, thank you, Robbie. So I know it's great to have you all on board listening to our webinar, and it's great for your support. And I'm very excited to review some recent data, as Robbie mentioned, that's just been released as part of our biological validation of the hydrogel to make sure it actually is suitable for the applications that we want to it be used for. So we've been working with our partners. The first thing we looked at was, well, is Jellagel non-toxic, which we've proven the case to be. So based on cytotoxicity, it's very biocompatible. As you can see there on the right-hand side, it is as biocompatible, slightly more than bovine collagen solution, which is great news. The first experiment we looked at was growing human-derived bone marrow mesenchymal stem cells for a competitor evaluation against Matrigel. As you can see here on the 3D confocal image here, the left-hand photograph, sorry, is showing very good distribution of mesenchymal stem cells in our gel matrix, and in a like-for-like study with Matrigel was really struggling on that like-for-like analysis. But this gave us very good first indications that the hydrogel was good for this particular application. Next slide, please. Looking at in more detail, obviously that was a 4-day analysis. Then we looked at for the gel for a longer period of time in these mesenchymal stem cells, comparing against Matrigel. And again, what we found was that in the like-for-like study comparing against Matrigel, we found that the Jellagel successfully supports the increased proliferation of viable human-derived MSCs from bone marrow samples, which is great news. So the product does enable the microenvironment proliferation of that cell line. Again, imaging you can do, and again with other partners, and this next slide was done with National Physical Laboratory, because obviously imaging of the cells in the gel is very important. And again, confocal, because we don't use phenol red in the product, it's a very clear gel, it enables very good imaging analysis to be done. So the next slide, when we look at this in more detail with our partners at the National Physical Laboratory, we did look at growth proliferation over 7 days again, but in a much more magnified analysis. And we showed that those mesenchymal stem cells were also showing their phenotypic characteristics as well, very fibroblastic in their morphology, which again is really exciting. And again, very biocompatible, very high cell viability, and very good proliferation over 1, 5, and 7 days. But what's good here is that that the cells within the gels can be fixed, they're permeabilized for the purpose of cell staining, and you get good healthy spreading of cells. In fact, one of the lead researchers on who did that study, Dr. Niloufar Faraki, she again exclaimed the ease of use of this protocol. It was very good to handle, and she was very pleased with it. So again, so for mesenchymal stem cells, it's a very good matrix, and this gave us very good excitement and very good encouragement that the matrix is very good. So the next slide, again, building on the cell lines and potential downstream applicabilities of this gel matrix, we looked at growing cancer spheroids in the matrix, and we looked at MCF7 breast cancer cell lines as an example. Now, we used the MCF cell line, the cell type for breast cancer, but the results in this micrograph are very encouraging. These cell lines like to form spheroids in a good microenvironment, so they will do that in Matrigel and other matrices. So it's very important that we see similar morphologies from the cell line, and in fact we do. And not only that, you see a very good structure of the cells within the spheroids that are indicative of growth, and also the cytoskeletal network is very evident as well, showing a very good, well-developed structure. So why is spheroids important? Well, this is showing indicative cancer-like morphology, and again should enable downstream drug modeling for various drug discovery or patient-derived precision medicine type approaches. So again, this has given us very good indicative excitement that we have the Jellagel matrix. It has a very good microenvironment for 3D cell culture. Okay, the next slide. So the next slide our cells. We can show that we can grow cells in spheroids. We can show that they're uniformly spread throughout the matrix. So we looked at their stability in vitro as well. And for this particular study we did with the Roslin Technologies up in Edinburgh, we looked at a cell line, the mouse embryonic stem cells. And again, the Jellagel supported the growth of these stem cell lines in good, robust clusters. And when we looked at stability of the phenotype, we stained them with alkaline phosphatase, which is a standard protocol that shows that maintenance of the cellular phenotype of which was proven in this study. So again, we're seeing spheroidal type growth. That spheroids are uniformly spread throughout the gel matrix, and they're stable in terms of their phenotype, which again is a very important feature of what we have here. And the next slide. Rob, do you want to do this? Robbie, you want to do this? Or I'm very happy to continue.
Yeah, no, that's fine. Rob, I think you were going to do this one, but I might as well. So thank you, Andrew, for sort of talking us through some of that data. Hopefully it does go to show just how good Jellagel can be. And a lot of it, as I say, is brand new. We've only had it in the last week or so. So yes, it's an honor to be able to present that today to such a number of people. So just to recap some of the benefits, it's a very easy to use hydrogel kit. Use it at room temperature. There's not really any deviation from standard lab protocols other than creating our gel. It's biochemically simple, and there's no unwanted or undefined growth factors in there. With products such as Matrigel, you do have a lot of undesirables in there. It is batch to batch consistent. In terms of the collagen content, you know that's exactly what you're getting. It's non-mammalian, it's disease vector free. There are no translatable diseases between jellyfish and humans. So when you're growing human cells, it's a great product to have. It's inert, cleaner at microRNA level. As the graph showed, it is much better in terms of toxicity as well. Phenol red free. So with that, you get no undesirable biochemical effects, but also it's great for imaging as well. And as a company as well, we're very proud to be ISO 13485 certified. As quite a small company, it's a great achievement for us to get that, and it hopefully will give customers like yourselves a real peace of mind that the product is coming from a very secure and controlled environment. So I will hand the mic over to Rob just to wrap up.
Thank you, Robbie, and thank you, Andrew. We do actually have one question that's just come in from Vanessa. So Vanessa's asking, did we try the Jellagel with neutral stem cells in order to obtain neurospheres?
Thanks, Rob. To answer that very clearly, we haven't tried Jellagel for that 3D neurospheres yet, but it is very possible because we have done an awful lot of work growing neural stem cells in 2 dimensions and get both co-cultures of astrocytes and cortical neurons in co-culture, showing some very good results in 2 dimensions. And again, that was compared against Matrigel, and what we did see was a much improved neuronal firing network, which was a lot more synchronous than it was on Matrigel. So it would be very encouraging to see what that would do in 3D, certainly. But the potential for that to be the case is very high.
Thank you, Andrew. We have another question from Elizabeth. Did you try to differentiate the BM-MSC towards osteoblast lineage, for example, and get mineralized matrix?
No, we haven't yet, but it is on our list of to-dos in terms of the characterization work that we're doing that's ongoing at the moment for MSC.
Okay, thank you. Chloe has asked, do you think it's possible to realize co-culture within this gel, spheroid and LTs, for example?
Absolutely. Again, we've shown that can be the case in 2D. So growing the co-culture in 3D with our Jellagel, it should be feasible. Absolutely.
Thank you. And the next one from Nicola, is it possible to use Jellagel to embed fresh acute slices, e.g. brain, spinal cord, in the perfusion chamber?
I don't know the answer to that. It is very possible. Again, own experimentation will find that out. But again, it shows a very good biocompatible matrix for the culture of cells broadly. So I don't see why that wouldn't be an issue at all.
Okay, thank you. Zamina, I hope I pronounced your name correctly. What is the highest concentration that we've done of Jellagel? So,
well, we do it at 4 mg per mil at the moment.
At the moment, yes, exactly. There are possibilities for work to be done on it, but everything that's done outside of our current offering would obviously be on an OEM basis, and it would require further discussions to see the reasoning behind you needing different concentrations or so on. So that is a discussion that can definitely be had as well. Question for Barbara, did you try to stain the DAB in immunohistochemistry?
DAB. Sorry, DAB. Sorry,
we've also missed a question from Elizabeth there, Rob.
Sorry.
Just Andrew, while you're looking for DABs, did we try it with endothelial cells to try and get endothelial tubes?
Not at the moment, no. But again, I understand that's a clear use of the Matrigel protocols in the past. But again, it would be very interesting to find out. Again, as long as the matrix is able to culture those cells, then absolutely, I don't see why there wouldn't be an issue.
Okay, thank you.
Thank you. I'm just
yeah, no worries.
the answer to that is not yet, no. So that question to the immunohistochemical staining.
No, okay. Elm has asked about use of Jellagel for bioprinting.
It's a very interesting area. It's something that we also have an interest in ourselves. As a matrix, as a collagen, irrespective of where you get it from, is very challenging to bioink as a native polymer. It's going to need a strategy of polymer chemistry to really optimize its strength and properties to maintain its strength, but also to maintain its biocompatibility for any ink application. So that would be a very much research project.
Thank you. Another one is, can Jellagel be used for organoid culture?
Well, we've seen very good results for spheroids, and the next, I guess, with co-culture, you could certainly look at that. We haven't done that yet, and again, but there's no reason, again, to believe that Jellagel couldn't assist in that endeavor.
Definitely. Can you inject Jellagel in an animal model?
Well, absolutely. So we have actually conducted in vivo experiments with different formats of our Jellagel. Remember, is composed of jellyfish collagen type 0, and we have implanted jellyfish collagen type 0 in other formats into various animal models of disease, and have shown very good biocompatibility, and in terms of tissue engineering, some very interesting results as a consequence of that, supporting it as a tissue engineering device. As a gel being injected, then I have no issues whatsoever. It should be fine.
Okay, good. Is there
there is a study that's been published. If people would like to know, we can share that publication reference if people want to know more information on that.
Yeah, certainly. Is there a possibility to isolate the cells afterwards by digestion, etc.?
Absolutely. We isolated this yes, actually through Liberase, and we do have a protocol for Liberase digestion of the gel to isolate the cells that we can share as well. So we do it routinely ourselves.
Yeah, great. So from our tests now, how many days is it possible to maintain cell culture that we've seen so far?
We've taken it to 7 days, but we're looking at longer term evaluation at the moment as well.
And then obviously the one below is for how long can you maintain cells in Jellagel with medium refresh?
Again, same thing. So we've seen 7 days, no problem. We are looking at extending it out to 14 at the moment. But again, seeing no signs of any issues at the moment.
Just one just popped up from Julia saying, is it possible? It is compatible with silicone plates.
Let me come back to you on that one. I need to check.
Yeah.
Yes, the digestion protocol is available, and we can put it on our website, or we can share it with distributors, no problem.
Okay, that's all of them so far. I'll just keep a second to see if any more pop in. Yep, is this gel thixotrope, e.g., can you resolubilize it after gelification? I think you've answered that one, haven't you?
Yeah.
Can we just have a recap of that question, just in case anyone missed the answer?
Okay, is the gel thixotrope? Can you resolubilize it after gelification? Okay, yeah. Through mechanical, I guess through mechanical agitation, you can liquefy it again if that's what you're asking. Liberase again would cause it to become a liquid again or resolubilized.
Yeah. With all of these types of questions that are coming in, obviously that it's, as you can see, is very much a wide range of ways the product would be used by yourselves. So as a company, Jellagel, working alongside CliniScience, we're offering our services to you. So if you were to take the kit and order it through CliniSciences, then we are always available for you for when you're using them. So when you plan your testing or your trial of the material, then we can organize a call with you at that time. So then the conversation we're having is obviously more directed towards your test. It's very difficult to cover off everything in this type of call because there's so many different ways it's being used. So yes, we're more than happy to support you on a technical point of view and make it more relevant to your specific test. And that being said, I think there's no more questions coming in. I think that is it from a point of view of the presentation for Jellagel. I hope you all have a better understanding of the product and its properties and how it can be used. Please, if you do wish to try the kit, like you said, it all comes as one whole kit, then please get in touch with CliniSciences. If you need their contact details, then I'm sure Florence will pass all of that on to everyone from this webinar. But again, thank you very much for your time. We really appreciate you joining our call today and learning a bit more about Jellagel.
Thanks a lot for this presentation. The webinar has been recorded, so the video will be available on our website soon. And of course, if you have any question, you can contact Clinisciences and all the subsidiaries in the different countries. We will be more than happy to help you and to answer to your questions. I will send all the contacts to all the persons in order that you can share your question with us.
Okay.
Thanks a lot, Jellagel team, for this presentation, and have a nice afternoon, everyone. Bye-bye.
Thank you.
Thank you all.